tables for clinical research.
Hi, thank you for having me. This package is developed by myself. I'm a lead statistician or lead data scientist at the Prostate Cancer Clinical Trials Consortium, and my co-developer, Daniel Soerg, is also on the line. He is a senior biostatistician at Memorial Sloan Kettering Cancer Center. So gtreg is a package that is built on gtsummary. That is a package that is built on GT, which means that all the tables you're going to see are highly extensible and customizable. As an overview of what's inside the package, we have some gtreg functions meant to table adverse events, and then we have other tabling functions as well. You can modify those tables using gtsummary modifications like modify header, and to specify how those modifications are enacted, you use gtreg column selectors. And lastly, your table can be exported to various devices like Word, PDF, and Excel. So let's talk about adverse events. An adverse event is a medical issue that occurs during the course of treatment or observation. And adverse events are classified according to our hierarchy. We typically report lower level terms within a system organ class, and we also record other features of the adverse event, like grade and drug attribution. So here you see 5 adverse events from a single patient recorded over time. But when we go to summarize this data, there's a lot of challenges. So first, subjects experience multiple adverse events over time. Not all enrolled subjects experience an adverse event, and the percent of subjects experiencing specific adverse events is of interest. So we typically count these by maximum grade per subject. And lastly, we have to do this a lot. So within a single reporting cycle, we might have to create multiple adverse event tables, like treatment emergent adverse events and adverse events on specific treatment cycles or serious adverse events. So what does it mean when I say we're counting by maximum grade? Here you see our 5 adverse events. And these adverse events are really 3 unique terms. 1 for anemia, 2 for increased tendency to bruise, and 2 for thrombocytopenia. So when we count by maximum grade, we're just going to be counting the highest grade observed per event per subject. And similar logic applies when counting by severity. So if mild, moderate, severe is stored as a factor, the highest factor level is retained. So let's get to your first adverse event table. We'll need some data. So the gtreg package comes with 2 example data sets. The first is DF adverse events, which has 10 unique subjects that experienced adverse events, but multiple rows per subject or multiple adverse events per subject. The second data set we have is DF patient characteristics, which has 100 unique subjects and 1 row per subject. Another feature that you might observe in our data is that our data has variable labels, which is an attribute of a variable in a data frame. And where applicable, variable labels and not variable names are going to be printed in our gtsummary and gtreg tables. You can set your own variable labels in your own data if you want to take advantage of this feature. And you can see if your data has variable labels by using the stir function or by viewing the data set in the RStudio viewer. So you'll see the variable name printed on top and the variable label printed underneath. So today we're going to be focusing on table AE, the main function I use to create adverse event tables. To create what table AE does is it counts adverse events per subject by maximum grade. So I'm going to take my DF adverse events data set, pipe it into the table AE function. I'm going to specify the unique identifier for a patient with the ID argument, the adverse event term with the AE argument, the system organ class term with SOC argument, and how I want to summarize that. In this case, I'm doing it by grade. The resulting table is going to look like this. And recall, DF adverse events has 10 unique subjects in it, which is why our subject denominator is 10. So here we can see that 7 out of our 10 subjects, or 70% of them, observed blood and lymphatic disorders, system disorders of grade 5. What you also notice about the organization of this table is that we have a higher level organization of our system organ class, and then lower level terms are indented underneath that. But we know from the previous discussion that we actually had 100 subjects of interest in this fictional data set. So we need to correct for that in our tabling. The way we're going to correct for that is we're going to supply IDDF to achieve the correct subject denominator. So IDDF means it's your ID data frame. What that looks like is that you're actually using 2 different data sets to compute this table, the DF adverse events and the DF patient characteristics. And what's important is that your unique patient identifier has to be the same between the two data sets. Now, when you supply table AE with the ID DF, the resulting table is corrected with the correct denominator of subjects at risk, which in this case is 100. So 7 out of 100 subjects, or 7% of your subjects, experienced grade 5 blood and lymphatic system disorders. We can also do tabulation by strata by including the strata argument. And the way the table will look is simply by separating out the adverse events by grade in strata now. Of course, people always want to modify and customize their adverse event tables. So here's a few ways you can do that. The first thing I'm going to do is add an overall column using the across argument and the by value. And we're also going to bold labels. So what this does is it adds an overall column summarizing across all by values, which in this case is grade, underneath each strata. There's actually several different ways you can add overall columns, so please look at the functionality or the documentation for that functionality. We've also bolded labels here, which in this case will bold our system organ class values to make them pop. Other ways people might want to modify this table is to modify how the header looks. So in this case, you see 1, 2, 3, 4, 5 without any context about what it means. And you see the treatment regimen and the sample size on the same line. You can use the modify header and modify spanning header functions to adjust this. So in this case, now I've modified it to print out grade in addition to the numeric value, and as well as putting the sample size on a new line underneath the treatment regimen. But again, due to the brevity of this talk, I'll refer you to the articles to learn more about how to actually implement this. We do have other tabling functions. So for example, table reg summary and table listing. Table reg summary is going to create summary tables with standard regulatory formatting. And by that, I mean when we have continuous variables, there's going to be a multi-line continuous summary automatically produced. And then your categorical variables are going to be summarized as usual. We also have table listing, which really isn't about summarizing data. It's about printing raw data, but printing it in a specific way with group terms. So this figure that you saw earlier about how to count by maximum grade, this is actually produced by table listing, where our terms are grouped under term 1, term 2, and term 3. But what's kind of neat about this table is that we're showing how the extensions work. Because gtreg is built on gtsummary, is built on GT, you can actually leverage GT functions to modify your tables. So when we get under the hood, this is a table listing, but you can convert it to a GT table and then use GT functions to implement the highlighting. We can also create table shells with this package using the style XXX function. And so here you're going to be creating generic table shells based on your table AE or your table reg summary. And you can see more information in our package down articles. Lastly, there's various ways to get these tables back out of R. HTML is the default output format. You can export to Word leveraging Flex Table, Excel using Huxtable, and PDF using Cable Extra. And when you go back and forth between these various outputs, for the most part, all of the formatting is retained. So if you want more information about gtreg, you can find it on our website. This package is on CRAN, so you can install it from CRAN or the development version from GitHub. If you have any issues with the package, please file that on our gtreg issues. And if you have any feature requests, questions, discussions, please pop over to our GitHub discussions board. We'd be happy to hear from you. Thank you.